Swayam Prabha. Digital India. Educated India.
We will continue our lecture series on the regulation of eukaryotic gene expression and in the last two lectures we have been trying to understand how molecules which either interact with membrane receptors or which enter into the cell and interact with soluble receptors ultimately transduce signals to the nucleus, leading to alteration of gene expression. To begin with, we have been looking at the membrane receptor signaling. In the last two lectures we have actually covered how molecules which are water soluble, therefore cannot penetrate the lipid barrier, interact with specific membrane receptors, and then how these molecules transduce signals leading to activation of gene expression. To begin with, of the various kinds of membrane receptors, we have been focusing our attention on one particular group of receptors, namely the G protein coupled receptors, which are 7 transmembrane receptors or serpentine receptors, to which when specific small molecules like epinephrine or even peptide molecules like insulin binds, it results in the activation of specific G proteins, which are trimeric G proteins, and these trimeric G proteins then get activated, and as a result the alpha subunit dissociates, and in one case it goes and activates adenylate cyclase, leading to activation of cyclic AMP, synthesis of cyclic AMP. Cyclic AMP then goes and activates protein kinase A, and protein kinase A now goes inside the nucleus and phosphorylates transcription factors like CREB, leading to activation of cyclic AMP responsive genes. So this is one pathway that we have studied in the last class. Now cyclic AMP is not the only second messenger which is synthesized in the cells. There are a number of other second messengers as shown in this particular cartoon. We have cyclic GMP, we have inositol triphosphate, diacylglycerol, calcium. All these things serve as very important second messengers in transducing signals into the nucleus and activating specific gene expression programs. So what we will do today is to look at regulation of gene expression by second messengers other than cyclic AMP. So let us take a look at and then see how molecules like cyclic GMP, inositol triphosphate, diacylglycerol and calcium, when they get activated, or how they go ahead and influence gene expression programs. That is going to be the crux of today's lecture. So what I just summarized what we have discussed in the last two classes. When we began our lectures on signal transduction pathways, we started with a very brief introduction about signal transduction. We discussed briefly about lipophilic signal molecules, how they diffuse through the cell membrane and interact with intracellular receptors, and how water soluble ligands, which since they cannot penetrate the lipid barrier, they interact with membrane receptors and activate signal transduction pathways, and there are a wide variety of membrane receptors with which these molecules interact, and we discussed about one particular class of membrane receptors, namely the GPCRs or G protein coupled receptors, and when these G protein coupled receptors are activated, they in turn activate trimeric G proteins, and the G alpha subunit now goes and activates adenylate cyclase, leading to the synthesis of cyclic AMP. Cyclic AMP in turn goes and activates protein kinase A, and then cyclic AMP and protein kinase A via the ATF family of transcription factors activate genes which are in response to cyclic AMP, which contain cyclic AMP response elements in their promoters. This is what we have discussed in the last two classes. So today let us see how regulation of gene expression is carried out by cyclic GMP, inositol triphosphate, diacylglycerol and calcium. Now this is what we have discussed so far, how when a hormone binds to a GPCR, it results in the activation of a G protein, which is a trimeric G protein, and the G protein now binds to GTP and gets activated, and in the GTP bound form it now goes and interacts with adenylate cyclase, and adenylate cyclase is now activated, and adenylate cyclase will now convert ATP into cyclic AMP, and this cyclic AMP thus synthesized now activates protein kinase A, and protein kinase A in turn activates phosphorylates molecules like CREB, and then resulting in activation of transcription. So this is the summary of the last class about how binding of a hormone molecule to a cell surface receptor results in the synthesis of cyclic AMP, leading to the activation of protein kinase A. Now I also discussed that we have discussed a very small part of cyclic AMP signaling, because our focus is primarily on the regulation of gene expression. Cyclic AMP, once synthesized, does a number of other things like activation of metabolic enzymes and number of other pathways, but we focused our attention primarily on regulation of gene expression. So, we confined ourselves to only one part of this complex pathway as shown here, namely how cyclic AMP once activated, how it activates protein kinase A, and then how protein kinase A goes inside the nucleus and phosphorylates a number of transcription factors leading to activation of gene expression. We have not really discussed a number of other things that cyclic AMP does, because we are not really we want to focus primarily on the regulation of gene expression by the second messengers, and therefore confined ourselves to only one particular aspect of this signaling pathway. I also focused primarily my attention in the last class on how protein kinase A activates ATF CREB family of transcription factors, and I have shown clearly that pKa phosphorylates serine 133 of CREB, and it also phosphorylates serine 44 of the ATF2, and as a result both of them now get activated, bind to cyclic AMP response elements and activate transcription of gene. We also discussed about how some of the mutants, some of the variants of the CREB which arise because of alternate splicing molecule like ISER, which lack the transactivation domain, when they get activated in response to pKa from the P2 promoter of CREM, they actually act as a negative regulators of genes. Now, more and more such transcription factors which are being regulated with pKa are being discovered. For example, in addition to the ATF CREB family of transcription factor, a protein called ER81 ETS is also has been shown to be phosphorylated by protein kinase A, and therefore this also plays a very important role in number of physiological processes. And similarly, another transcription factor called HNRNPA1, which plays a very important RNA processing. Again, this is also a target for pKa. It gets phosphorylated on serine 199. That also is a is activated by pKa signaling. So, although we focus our attention primarily on the ATF CREB family of transcription factors which are the target for cyclic AMP and pKa, more and more transcription factors are being recognized as targets for pKa and are being identified and being studied. So, having studied the cyclic AMP dependent protein kinase A pathway, today's lecture will focus primarily on cyclic GMP dependent protein kinase G pathway and how calcium and calmodeling kinase as well as diazyl glycerol activates another protein kinase, very important protein kinase called protein kinase C, and how and this will lay the foundation for us to discuss about how protein kinase C once it is activated, how it goes and regulates gene expression programs. So, in this particular cartoon, I have basically summarized what all we are going to discuss today and what we discussed yesterday in the last class. For example, there are three major signaling pathways that we are going to focus on in the last class and today's class. What we have discussed so far is the activation of membrane receptors which activate trimeric G proteins, in which the G alpha subunit interacts primarily with adenylate cyclase, and as a result ATP is converted to cyclic AMP, which then activates protein kinase A that now goes inside and phosphorylates a number of transcription factor leading to activation or repression of gene expression. This is what we have discussed now. What we are going to discuss today is about a receptor which actually possesses intrinsic guanylate cyclase activity. Okay. Now, in this case the trimeric G protein interacted with the adenylate cyclase leading to the activation of synthesis of cyclic AMP, but the cyclic GMP synthesis actually involves a membrane receptor which has an intrinsic guanylate cyclase activity, and as a result the binding of a ligand to the receptor activates its guanylate cyclase activity, and as a result this guanylate cyclase now converts GTP to cyclic GMP, which then activates protein kinase G and which then goes inside and then phosphorylates transcription factor leading to activation of gene expression programs. So, this is one pathway we are going to discuss now. The other thing we are going to discuss is molecules like neurotransmitters which and certain hypothalamic hormones, they activate a different set of G proteins which just like this G protein here activates adenylate cyclase, and here you got activation of the guanylate cyclase, this particular G protein now activates phospholipase C, and phospholipase C now converts phosphatidylbis inositol phosphate into inositol phosphate 3 and diacyl glycerol, and these two are very important second messengers. Inositol triphosphate now goes and releases calcium from the endoplasmic reticulum, and calcium thus released now activates the calcium calmodeling kinase, and this calcium and calmodeling kinase then goes and activates the cAMP kinases or calmodeling protein kinases, which again goes and either phosphorylates a number of enzymes or proteins either outside the nucleus or transcription factors inside the nucleus, leading to activation of specific physiological processes. The diacylglycerol also is a very important activator of protein kinase C. Both calcium and diacylglycerol together can also activate protein kinase C, and protein kinase C again can phosphorylate a number of proteins including transcription factors, leading to activation or repression of target genes. So, these are the three major signal transduction pathways that we are going to discuss today, as well as yesterday, that in the last class we have covered, which involves trimeric G proteins and how some of these trimeric G proteins activate adenylate cyclases and some of the G proteins activate phospholipase C. In the process, second messengers like cyclic AMP, cyclic GMP, inositol triphosphate and diacylglycerol, all the second messengers synthesized which amplify the signal, go and then phosphorylate a number of enzymes or proteins including transcription factors, leading to alteration of gene expression. So, let us now see how does calcium regulate gene expression. What is the important features of calcium signaling? Now, we have already discussed in the previous class how calcium can also activate CREP. So, CREP can be activated not only by cyclic AMP, CREP can also be activated by multiple signaling pathways. That is what we discussed last class, and one of them is calcium. So, once the calcium enters the cell, calcium activates calmodulin, or calcium can also released by from the endoplasmic reticulum. Once the intracellular calcium, cytosolic calcium increases, calcium now activates calmodulin, which is a very important protein, and calmodulin in turn activates CAMP kinases, and CAMP kinases phosphorylate multiple targets including serine 133 of cyclic AMP responsive binding protein. In fact, we discussed in detail in the last class how the CAMP kinases, especially in the nervous system, play a very important role in the activation of CREB, and how cyclic AMP as well as CAMP kinases activate CREB, and in turn it involves in the long term memory and so on and so forth. So, let us now see, in addition to activation of CREB, how does calcium influence gene expression? That is what is going to be the focus of today's class. Now, there are two different mechanisms by which calcium can activate gene expression programs. One is by what is called as the diacylglycerol dependent protein kinase C pathway, and another is using the CAMP kinase pathway, which we just discussed. So, let us now, this is a brief schematic that actually gives you the crux of what we are going to discuss now. Once the hormone binds to a specific cell surface receptor, it results in the activation of a G protein, trimeric G protein, and this G alpha subunit of this G protein now activates phospholipase C. So, in the case of the cyclic AMP pathway, the G proteins, the G alpha S subunit activates the adenylate cyclase, and as a result you got the synthesis of cyclic AMP, but here the G protein activates phospholipase C, and as a result you get the synthesis of two different second messengers, namely inositol triphosphate and diacylglycerol. Now, phosphatidylinositol bisphosphate is a very important lipid molecule that remains anchored to the cell membrane, and phospholipase C now cleaves the phosphatidylinositol bisphosphate into inositol triphosphate and diacylglycerol. Now, the inositol triphosphate thus synthesized now goes into the endoplasmic reticulum, and then mobilizes the calcium release, causes calcium release, and now the calcium release now goes and activates the protein kinase C along with diacylglycerol, and the protein kinase C thus activated now phosphorylates a number of enzymes, proteins including transcription factor, leading to specific physiological methods. So, calcium by virtue of activating leading to synthesis of diacylglycerol and calcium release and activation of protein kinase C can influence specific gene expression programs. So, the protein kinase C that is now synthesized now can go and phosphorylate a number of enzymes or proteins including transcription factors, resulting in specific biological effects. So, this is one pathway by which calcium can activate gene expression programs. And as we already discussed, the calcium, when the same hormones or ligands bind to specific receptor activate gene proteins, the phospholipase C that is activated now results in the mobilization of formation of inositol triphosphate, which mobilizes the calcium store. Now, the calcium that is now released in the last slide we have mentioned how it goes and activates protein kinase C. The same calcium can also now combine with calmodulin, and then the calcium calmodulin now can bind to calcium on binding to calmodulin. In presence of calmodulin, the CAMP kinases get activated, and these CAMP kinases now goes and phosphorylate a number of enzymes and proteins, leading to specific biological effects. So, calcium can activate both protein kinase C on one hand along with diacylglycerol, and calcium along with calmodulin can also activate CAMP kinases. So, calcium activates signal acts as a signal transduction molecule acts as second messenger in two different pathways. One is calcium together with diacylglycerol can activate protein kinase C, which can phosphorylate a wide variety of substrates, leading to activation and repression of specific gene expression programs. The calcium can also combine with activate calmodulin, and calmodulin now can activate cAMP kinases, and cAMP kinases can activate different set of proteins and transcription factor, leading to specific biological effects. Now, what is calmodulin? Calmodulin is a calcium binding protein, and 4 calcium ions can actually bind to 1 molecule of calmodulin, and the calcium calmodulin complex now goes and binds to calcium calmodulin kinase and induces a conformational change in the calmodulin kinase, and now the calmodulin kinase now can go and phosphorylate specific serine and threonine residues of target proteins, and as a result one of the physiological effects that happens in the cAMP kinases is the increase in the intracellular calcium levels, especially from the endoplasmic reticulum. Calcium is released in the cytoplasm. There is an increase in calcium reserve inside the cytoplasm, and it can also result in the activation of adenylate cyclases, guanylate cyclases, and so on and so forth. So, a number of physiological processes like glycogen synthase, phosphorylase, kinase, etc. are all activated by cAMP kinases, and there are also a number of transcription factors including CREB which are activated by cAMP kinases. So, there are 2 pathways we have discussed so far, especially involving the G protein coupled receptors. In one pathway, when a signaling molecule binds to the GPCR specific receptor, it now activates in the activation of a trimeric G protein, and then the alpha subunit, especially the GS alpha, G alpha S subunit activates adenylate cyclase, leading to synthesis of cyclic cAMP, which results in the synthesis of protein kinase A, and protein kinase A now goes and phosphorylates a number of transcription factors, leading to activation and repression of target genes. So, this is what is called the cyclic cAMP pathway. And we also now discuss just the calcium pathway, wherein with different kind of signaling molecules, when they receptor with their cognate receptors, that results in activation of a different class of G proteins, and here the G alpha subunit is called the GQ, which now instead of activating adenylate cyclase, here it activates phospholipase C, and phospholipase C now converts inositol diphosphate into diacylglycerol inositol triphosphate, and diacylglycerol and inositol triphosphate now mobily releases and results in the calcium release, and the calcium together with diacylglycerol now activates protein kinase C, and calcium also binds to calmodulin, resulting in the activation of calmodulin kinases, and these 2 kinases together can go and phosphorylate a number of proteins including transcription factors, leading to activation and repression of target genes. So, these are the 2 major signal transduction pathways we have discussed so far, of how cyclic cAMP and calcium are acting as very important second messengers. Now, let us now spend some time to understand, in addition to cyclic cAMP, there is also a very important cyclic nucleotide, namely cyclic GMP, and how cyclic GMP acts as a signaling molecule. Now, cGMP, as we just discussed now, is produced by guanylate cyclase, which catalyzes the conversion of GMP to cyclic GMP. Now, the guanylate cyclases that are actually in 2 forms, a membrane bound form and a soluble form. We will discuss this in just a minute. Now, what are the intracellular targets of the cyclic GMP? Once cyclic GMP is synthesized inside the cells, they can go and activate cyclic GMP dependent protein kinases. These are usually referred to as G kinases, and it can also activate cyclic GMP regulate phosphodiesterases, which actually cleave cyclic GMP, and cyclic GMP is also in opening up of specific ion channels, especially in the nervous system. So, cyclic GMP has a number of cellular functions, including activation of specific protein kinases, phosphodiesterases, as well as opening or closing of ion channels. Now, what I would like to now emphasize here is that whenever we talk about second messengers like cyclic AMP, calcium, or cyclic GMP, they perform a number of effects. So, what I would like to emphasize from this cartoon is that second messengers such as calcium or cyclic GMP regulate multiple cellular processes, and as I have shown here, they can affect cellular trafficking, they can alter the enzyme activity, they can either activate enzymes, they can inhibit enzymes, they can inhibit protein synthesis, they can alter ion channels, so the outflow and inflow of ions can be affected. But since in this particular course we are focusing our attention primarily on regulation of gene expression, we are not going to discuss some of these aspects. So, we are going to focus primarily on how second messengers regulate transcription inside the nucleus, and therefore, we are not going to discuss many of these aspects. So, when I say this is what second messengers do inside the cell, please be aware that second messengers also do a number of other things. But since our focus in this course is primarily on regulation of gene expression, we are not going to discuss many of these aspects. So, we are going to focus primarily on how binding of a ligand to a cell surface receptor resulting in the synthesis of second messengers, and how the second messenger go and influence gene expression program. This is what we are going to focus, and we are not going to discuss many other things that second messengers do outside the nucleus, but these are all very important processes, and they have a very important applications in a number of biomedical relevance, and many diseases actually manifest because of this role of second messengers in the activity in the activation and repression of a number of enzymes, including diseases like diabetes and so on and so forth, are all affected by this second messenger signaling. Now, let us now spend some time to understand how cyclic GMP regulates gene expression. As I just mentioned, when the ligand or the hormone binds to a specific GPCR, it results in the activation of a G protein, and this G protein, now the trimeric G protein, the alpha subunit separates, and this alpha subunit separate now goes and activates some of these receptors have intrinsic guanylate cyclase activity, and as a result this results in the synthesis of cyclic GMP. Now, there are actually two kinds of cyclic the guanylate cyclase receptors, and what we have been discussing so far is about the membrane receptor, and which actually results in cyclic GMP. There are also what are called as soluble receptors, and which actually are activated by molecules like nitric oxide and carbon monoxide and so on and so forth. Now, there are again there is a typical similar example we have studied in the case of the protein kinase A. Protein kinase A also exists both in the form of a soluble protein kinase A as well as protein kinase A in kind of an anchors proteins like AKAP anchors protein kinases and then keep it in a anchored form, and the soluble protein kinases protein kinase A perform a different kind of function, and the anchored protein kinase A perform a different kind of function. Similarly, the guanylate cyclase also there are two types, the membrane bound guanylate cyclase and the soluble guanylate cyclase receptors, and the molecules gaseous molecules like nitric oxide and carbon monoxide primarily activate the soluble receptors, whereas polypeptide hormones like the atrial natriuretic peptide or ANP, which actually plays a very important role in vascular dilation. So, has a very important role in cardiovascular disorders or regulation of blood pressure and so on and so forth. These peptides, when they bind to their specific membrane receptors, they activate the membrane bound guanylate cyclase receptors. Now, since the focus in our lecture series is primarily on the regulation of gene expression, we shall skip the activities of the soluble GC receptors and confine ourselves to how the membrane guanylate cyclase receptors influence gene expression programs. So, let us now try to understand how molecules like A and P, when they interact with the guanylate cyclase receptor, how it results in affects the gene expression. Now, ANP is a very important molecule, and when it binds to its receptor, it stimulates its intracellular guanylate cyclase domain. So, this is slightly different from what we discussed so far. The earlier discussions of activation of the guanylate cyclase, when the ligand binds to the receptor, it activates a trimeric G protein, and this G protein, now the G alpha subunit goes and activates adenate cyclases. In the case of the cyclic GMP pathway, the hormone directly binds to receptor which has intrinsic guanylate cyclase activity. Okay, and this guanylate cyclase activity is activated when the hormone binds to the guanylate cyclase receptor, and the ANP receptor has been named natriuretic peptide receptor 1 or guanylate cyclase A, which is a 130 kilodalton transmembrane protein which is capable of converting GTP to cyclic GMP. The active GCA receptor is a homodimer containing an extracellular ANP binding domain at its amino terminal domain to which the ligand binds, and an intracellular guanylate cyclase domain at its carboxy terminal domain. So, once the ligand binds to the extracellular domain, it causes a conformational change, and as a result the cytoplasmic guanylate cyclase domain is activated, and that now converts GMP into cyclic GMP, GTP to cyclic GMP. Now, the cyclic GMP molecule is thus synthesized, bind to specific target proteins including cyclic GMP dependent protein kinases 1. There are about 2 different kinds of kinases called PKG1 and PKG2, and the cyclic include gated ion channels, and the cyclic may be phosphodiesterase and so on and so forth. So, the cyclic GMP has a number of targets including the kinases, the gated ion channels as well as nucleotide diphosphatase, which actually cleave the cyclic GMP back to the inactive form. So, on activation by cyclic GMP, PKG phosphorylates proteins such as CREB ATF1, which in turn activates expression of various target genes. So, this is basically the mechanism by which when a hormone like when a molecules like A and P bind to the receptor, which are nothing but guanylate cyclases, which has intrinsic guanylate cyclase activity, binding of this hormone to this receptor activates their guanylate cyclase activity, resulting in the synthesis of cyclic GMP, which then activates a specific kinases, influences ion channels as well as specific phosphodiesterases. Now, transcription regulation by cyclic GMP can also occur indirectly. The indirect control affects upstream signaling pathways modulating specific targets. The previous slide we actually discussed the mechanism by which cyclic GMP directly activates cyclic GMP kinases or phosphodiesterases. In turn, they target transcription factors and activate or repress gene expression programs. Now, there is also indirect method by which cyclic GMP regulates gene expression. For example, cGMP can activate mitogen activated protein kinases or MAP kinases, which regulate several transcription factors such as CREB, ternary complex factors, ATF2, cJun, etc. So, the cyclic GMP has two different roles as far as the gene expression is concerned. In one case, the cyclic GMP activates specific kinases like cyclic GMP kinases or protein kinase G, which then phosphorylate transcription factors leading to activation or repression of transcription. In indirect matter, the cyclic GMP goes and affects the activity of MAP kinases, and the MAP kinases in turn affect the activity of transcription factors like CREB or cJun or ATF2, resulting in activation of different set of genes. So, depending upon whether the cyclic GMP is activating protein kinase G or the cyclic GMP is activating MAP kinases, different set of genes are getting activated or repression, and you get different physiological effects. So, this is basically a summary of what we have discussed so far about the cyclic GMP dependent pathway. There are two different receptors where, likewise I said, there are soluble guanylate cyclases and there are receptor linked guanylate cyclases. Peptide hormones like peptide molecules like the ANP actually activate the receptor linked guanylate cyclases, leading to synthesis of cyclic GMP, whereas gaseous molecules like nitric oxide and carbon monoxide, they activate the soluble guanylate cyclases, leading to synthesis of cyclic GMP. And once cyclic GMP is synthesized in the cells, it is in the activation of protein kinase G, which goes and phosphorylates a number of enzymes and proteins including transcription factors, manifesting in the form of specific biological effects. So, again, once the cyclic GMP is synthesized by the guanylate cyclases either by ANP or through nitric oxide, one of the major activities of cyclic GMP and protein kinase G is to induce vascular dilation, which has a very important clinical effects, and there are a number of drugs and molecules which are actually used to cause vascular dilation, and they all affect through this particular pathway. So, cyclic GMP pathway plays a very important role in vascular dilation. The intracellular cyclic GMP concentrations are regulated by the action of guanylate cyclases and by the rate of degradation by cyclic GMP specific phosphodiesterases. So, just as in the case of the cyclic cAMP, we have seen that protein kinase A is an activated positive modulator of cyclic cAMP. At the same time, the cyclic cAMP is actually the protein phosphatases actually inactivate the protein kinase A and then make the cyclic cAMP signaling. There are phosphodiesterases which specifically cleave the cyclic AMP. In the same way, the cyclic GMP concentrations are regulated both by activation of guanylate cyclases which synthesize increase cyclic GMP concentration, whereas once cyclic GMP is synthesized, they are degraded by cyclic GMP specific phosphodiesterases. I am emphasizing this point very important because not only the adenylase guanylate cyclases which increase cyclic GMP concentrations, the GMP specific phosphodiesterases also has a very important physiological functions. For example, nitric oxide activates soluble guanylate cyclase, which in turn catalyze the formation of cyclic GMP from GTP, whereas cyclic GMP specific phosphodiesterases catalyze the hydrolysis of cyclic GMP to GMP, ending the signal transduction. So, on one hand, when you activate the guanylate cyclase, the intracellular concentration of cyclic GMP increases. On the other hand, when you activate a phosphodiesterase, it cleaves the cyclic GMP, leading to a decrease in the cyclic GMP concentration, and both have very important physiological functions. I will give you one example. For example, phosphodiesterase 5 and phosphodiesterase 6 as well as phosphodiesterase 9 are very specific for cyclic GMP, and they specifically cleave the cyclic GMP and inactivate it. This has very important applications because one of the drugs they known as the sildenafil, or very popularly known as Viagra, which is it's actually a novel inhibitor of cyclic GMP specific phosphodiesterase 5, and as a result it increases concentration of cyclic GMP, and therefore this is very effectively used in the clinical management of diseases like erectile dysfunction. So, you can see inhibitors of these specific cyclic GMP phosphodiesterases can be used as very important drug molecules for treatment of specific physiological conditions. There are actually two specific papers I have quoted here which actually led to the discovery of this Viagra, and then discussed how it acts as an inhibitor of this phosphodiesterase, and as a result the cyclic GMP levels increases, leading to a specific physiological effects. Now, in the central nervous system, The nitric oxide sensitive soluble guanylate cyclase isoform is the major enzyme responsible for cyclic GMP synthesis. So, the soluble guanylate cyclase plays a very important role in the nervous system and it has a very important functions. And the phosphodiesterases catalyze the hydrolysis of cyclic GMP in the brain, and there are mainly 3 isoforms which I have shown in the previous slide. So, the nitric oxide cGMP signaling pathway has been shown to play a very important role in the processes underlying learning and memory, and some of these things also involves activation or repression of specific genes. Similarly, aging is associated with an increase in phosphodiesterase expression and activity, and as a result a decrease in the cGMP concentration. So, in the nervous system the cGMP plays a very important role activating a number of physiological processes, especially the soluble guanylate cyclase play a very important role. So, having discussed 2 important signaling molecules, namely the cyclic cAMP and cyclic GMP, let us spend some time to understand how lipid molecules such as the inositol triphosphate, diacylglycerol, they serve as important second messengers. We have already discussed, but I would like to re-emphasize again because these are very important second messengers. Like we discussed earlier, when a hormone binds to the cognate receptor, it resists the activation of a specific G proteins, and this trimeric G proteins, now the alpha subunit now it activates phospholipase C, and as a result the phosphatidyl inositol bisphosphate is now converted to inositol triphosphate as well as diacylglycerol. So, PIP2 is converted to diacylglycerol and IP3 by phospholipase C, and this diacylglycerol now goes and activates protein kinase A along with calcium. Now, once IP3 is synthesized or the inositol triphosphate is synthesized, inositol triphosphate goes and interact with specific receptors in the endoplasmic reticulum, and as a result calcium is released from the endoplasmic reticulum. Therefore, cytosolic calcium levels rise, and calcium can either go and activate together with diacylglycerol can activate protein kinase C, or it can also bind to calmodulin, and the calcium calmodulin complex now goes and activates cAMP kinases, leading to specific physiological effects. So, the 2 important lipid molecules which plays a very important role in this entire pathway are the diacylglycerol and the inositol triphosphate. So, these 2 are very important second messengers. Hydrolysis of, just as I just mentioned, hydrolysis of phosphatidyl inositol bisphosphate generates diacylglycerol and inositol triphosphate, and diacylglycerol together with calcium activates protein kinase C, whereas inositol triphosphate is required in the mobilization of or increase in intracellular calcium. So, this is the 2 major functions of these 2 important lipid molecules. Now, what does protein kinase C do? Protein kinase C is again a very important regulator of number of metabolic processes, and protein kinase C is just like you have the calcium calmodulin kinase as well as protein kinase A is also a serine-threonine protein kinase. So, it goes and phosphorylates specific serine or threonine residues of a number of target proteins. And not only the PKC regulates a number of enzymes involved in various metabolic pathways, the PKC also plays a very important role in activation or repression of specific gene expression programs. So, PKC phosphorylates a number of transcription factors, and one of the important transcription factors which are phosphorylated by PKC is C-phos and C-Jun, which together are called as the AP1 or activated protein 1. Now, in the next class we are going to spend a lot of time trying to understand how phosphorylation of these transcription factor based protein kinase C results in the activation of very important physiological processes including cell proliferation and growth. So, just remember today's class that PKC plays a very important role in the phosphorylation of number of transcription factors which results in the activation of specific genes, and as a result activation of these genes results in proliferation of cell proliferation. Now, there are, in addition to this there are also a number of other lipid molecules which serve as second messengers, including for example phospholipase A2, which actually is in the results in synthesis of arachidonic acid, which again plays a very important role especially in a number of pathways involving prostaglandins and so on and so forth. And just now we discussed that phospholipase C synthesizes diacylglycerol and inositol phosphate and can also lead to the synthesis of ceramides, whereas phospholipase D is involved in the synthesis of phosphoric acid. So, the phosphatidyl inositol bisphosphate can lead to the synthesis of a number of lipid based second messenger molecules, And what I discussed so far is how phospholipase C converts phosphatidylinositol bisphosphate to diacylglycerol and inositol triphosphate. The same phosphatidylinositol bisphosphate can also be acted upon by phospholipase A2, resulting in synthesis of arachidonic acid, whereas phospholipase D converts phosphatidylinositol bisphosphate to phosphatidic acid, and all these things have a very important physiological functions. So, what are the cellular effects of some of these second messengers? Protein kinase C, some of these lipid molecules primarily activate the protein kinase C, and protein kinase C, as I just mentioned, plays a very important role in the alteration of specific gene expression programs. In addition, it also affects number of other processes which does not involve activation or repression of gene expression, such as cell motility as well as production of next generation of second messengers. A number of hormones actually act through this phospholipid breakdown. So, when you have these phosphatidylinositol molecules, these phosphatidyl molecules which are present in the cell membrane are now precursors for a number of second messengers, and when hormones like epinephrine, norepinephrine bind to alpha adrenergic receptors, or when acetylcholine binds to muscarinic receptors, or when TNF alpha, tumor necrosis factor alpha, binds to its TNF receptor, all these things ultimately results in the breakdown of phospholipids such as phosphatidylinositol bisphosphate, resulting in the generation of either activation of phospholipase A2 or protein kinase C or phospholipase D, resulting in the synthesis of specific second messenger molecules. And all the second messenger molecules have a very important physiological functions, and protein kinase C especially plays a very important role in the activation of transcription factors like CREB, c-Jun, c-Fos, etc., leading to cell proliferation. Now, the phosphatidylinositol bisphosphate breakdown is the second messenger system for the GQ family of G proteins. So, as I mentioned in the cartoon, the GQ is the G alpha subunit, and GQ actually specifically activates phospholipase C, whereas the GS subunit specifically activates the adenylate cyclase, leading to synthesis of cyclic AMP. So, there are hormone molecules which interact with specific set of G proteins and which contains the G alpha GS subunit, and this GS subunit interacts with adenylate cyclase, leading to the activation of synthesis of cyclic AMP, whereas in the case of the lipid molecule, the second messenger system, the GQ family of G proteins, where the alpha subunit is called as GQ, and this GQ, instead of interacting with adenylate cyclase, actually interacts with phospholipase C, activates phospholipase C, resulting in the conversion of breakdown of phosphatidylinositol bisphosphate into diacylglycerol and inositol triphosphate. So, GQ actually activates phospholipase C, and the phospholipase C is a membrane bound enzyme, and phospholipase C breaks down phosphatidylinositol bisphosphate into inositol triphosphate and diacylglycerol. So, you can see there are two different kinds of G proteins. One kind of G proteins containing the GS subunit interact with adenylate cyclase, resulting in synthesis of cyclic AMP. Another set of G proteins containing the GQ subunit, the alpha subunit is called as GQ, which interacts with phospholipase C, resulting in the breakdown of inositol bisphosphate into inositol triphosphate and diacylglycerol, which again act as very important signaling molecules. Now, the point that we would like to emphasize is that the breakdown of phosphatidylinositol bisphosphate results in generation of IP3 and diacylglycerol, both act as second messengers, while inositol triphosphate increases intracellular levels of calcium, resulting in the release from intracellular sources such as endoplasmic reticulum. Diacylglycerol primarily activates protein kinase C, and protein kinase C in turn goes and activates a number of phosphorates, a number of transcription factors and metabolic enzymes, bringing about specific biological effects. So, the protein kinase signaling is a very important signaling pathway, and so we are going to spend considerable amount of time in the next class to understand what are the major transcription factors which are phosphorylated by protein kinase C, and how phosphorylation of this particular transcription factors results in the activation or repression of specific gene expression, and how this manifests in the form of specific physiological effects. So, protein kinase C is a serine threonine kinase. It is activated by diacylglycerol as well as calcium, and it phosphorylates various cellular effects, and one of the major targets for protein kinase C signaling pathway is the transcription factors AP1, which consists of c-Fos and c-Jun, and both are proto-oncogenes. The levels of c-Fos and Jun are very finely controlled inside the cells, and if you overexpress some of these proteins, it can result in cancer. So, the PKC signaling pathway plays a very important role. For example, if you treat cells with molecules like 4-ball esters, which activate the protein kinase C, resulting in the synthesis of diacylglycerol and resulting in the activation of the AP1, it can result in cancer. So, in fact, the PKC activation is a very important animal model for understanding cancer, where if you treat mice, for example, continuously with molecules like Farbol esters, you can actually demonstrate the development of tumor right at the site of treatment. So, you can actually induce tumors in mice by simply treating with Farbol esters, which actually like TPA, tetraphorbinol tecnoic acid, which actually can cause activation of protein kinase C, resulting in the activation of AP1 and leading to cancer. So, PKC signaling plays a very important role in cell proliferation, and abnormal activation of PKC signaling can convert normal cell into cancer cells. In addition to the diacylglycerol and inositol triphosphate, which we discussed just now, there are a number of other lipid molecules which actually serve as second messengers for intercellular signaling. The IP3 and diacylglycerol we discussed so far basically serves as molecules second messengers for intracellular signaling. A number of lipid molecules also serve as second messengers for intercellular signaling. Some of them are listed here. These include eicosanoids, arachidonic acid derivatives, prostaglandins, thromboxanes, leukotrienes and lipoxins. And again, all these molecules are very important from the biomedical point of view, and they all control very important physiological processes, and a number of pharmaceutical companies are trying to develop molecules which either antagonize or which can stimulate the action of these, block the action of the second messengers. They have very important role in processes like inflammation, controlling pain and so on and so forth. So, this pathway, second messenger pathway involving lipid molecules has tremendous pharmacological importance, and all these molecules are very important from the pharmacological point of view, and a number of molecules which are available in the market either act as block the pathways involving the second messengers or activate the pathways involving second messengers, leading to specific clinical benefits. So, the lipid derived second messenger for intercellular signaling, they actually, once they are these second messengers synthesized in the cell, they escape from this cell where they are synthesized and participate in specific intercellular signaling. They sensitize normal physiological responses and they act as important local inflammatory mediators. So, you can see activation of this lipid based second messengers has a very important role, not only in controlling a number of physiological processes, but also for a number of therapeutic benefits. So, a number of drug molecules which either block or activate these second messenger pathways are used routinely in a number of processes like anti-inflammation agents and in pain management and so on and so forth. So, what we have discussed so far in the last three classes basically, we started discussing about how genes which are present inside the nucleus, where their expression has to be regulated by molecules which are present outside. And we discussed primarily how molecules which cannot enter the cell, especially the hydrophilic molecules or water soluble compounds, how when they interact with specific membrane receptors can activate specific kinases, leading to the synthesis of specific second messenger molecules, and these second messenger molecules in turn activate specific protein kinases, and they in turn phosphorylate specific transcription factors, leading to activation and repression of target genes. So far, among the various membrane receptors, we focused our attention primarily on one class of receptors called as the GPCRs or G protein coupled receptors. And these G protein coupled receptors are characterized by the presence of the serpentine receptors or the 7 transmembrane helices, and they act as receptors for a number of very important hormones like epinephrine, norepinephrine and so on and so forth. And we discussed very clearly how when these G proteins are activated, the G alpha subunit gets dissociated, and in one case this G alpha subunit goes and activates adenylate cyclase, leading to the synthesis of cyclic AMP. So, we have one class of receptors that regulate cyclic AMP formation, and the G subunits of these are called as the GS subunits, and there are also other kind of receptors which have an intrinsic cyclic adenylate guanylate activity, guanylate cyclase activity, and when these receptors are activated by molecules like ANF, ANP or atyl natriuretic peptide ANP, the cyclic JMP is synthesized because of the activation of the guanylate cyclase activity of these receptors, and this cyclic JMP in turn can activate a number of other physiological processes. Now, what we have also discussed today is that there are another kind of receptors which can activate a specific set of G proteins, in which the G alpha subunit, in this case it is called as the GQ subunit. Now, just as the GS subunit activates adenylate cyclase, the GQ subunit goes and activates the phospholipase C, and activation of this phospholipase C by this kind of class of G proteins now results in synthesis of diacylglycerol and inositol triphosphate from molecules like phosphatidylinositol bisphosphate, and this diacylglycerol and inositol triphosphate that is synthesized now act as very important second messengers. So, basically we have discussed so far about 3 different classes of G protein coupled receptors. In one case, the activation of this G protein results in the synthesis of cyclic AMP, in another case it results in the activation of synthesis of cyclic GMP, and in one more case it results in the synthesis of 2 important second messengers, namely diacylglycerol and inositol triphosphate. Now, in the what we have to discuss today is that GPCRs are not the only class of membrane receptors. There are also receptors which possess intrinsic protein kinase activity. There are receptors which have intrinsic tyrosine kinase activity, and there are also receptors which, when bound by a ligand, activate or recruit tyrosine kinases or protein kinases. So, we have to now discuss another class of membrane receptors which have intrinsic protein kinase activity or which can actually recruit protein kinases, and as a result a different kind of phosphorylation cascade is initiated, leading to activation and repression of target genes. Now, before we discuss how tyrosine kinases act as very important signal transducing molecules, especially for molecules like growth factors, and many of the growth factors like the EGF epidermal growth factor receptor, they possess intrinsic tyrosine kinase activity, and we have to now discuss how molecules like growth factors, when they epidermal growth factor, when they bind to specific receptors, the tyrosine kinase activity of this receptor is activated, and as a result a different kind of G proteins, in this case monomeric G proteins, especially the RAS family of proteins, they get activated, and you have initiation of a different kind of a MAP kinase cascade, leading to activation and repression of target genes. So, the activation of these tyrosine kinases involve activation of monomeric G proteins such as S, but so far our discussion has been primarily confined, especially with the GPCRs, we have confined our discussion primarily to G proteins which involve trimeric G proteins, and these trimeric G proteins are involved either in the synthesis of cyclic AMP or in the synthesis of diacylglycerol and inositol triphosphate. Okay. So, what we will discuss in the next few classes is how, when certain hormones, especially the polypeptide growth factors like epidermal growth factor, when they bind to the cell surface receptors, these cell surface receptors tyrosine kinase activity is activated, and as a result it now activates monomeric G proteins like RAS, and this RAS in turn activates a series of kinases, leading to the activation of the MAP kinase pathway, resulting in the activation and repression of specific target genes. But before we now go and discuss into tyrosine signaling pathway, I would like to discuss in the next class how the protein kinase C, which is what is the crux of the today's discussion, how once protein kinase C is synthesized or activated in the cells, how protein kinase C goes and activates specific transcription factors. Especially we are going to discuss how protein kinase C activates what are known as the early response as well as late response genes, and protein kinase C goes and phosphorylates proteins like activates molecules like CFOS and CJUN, and this CFOS and CJUN are called the immediate response genes or the early response genes, and this CFOS and CFOS-JUN again gets phosphorylated by protein kinase C, and these in turn go and activate the transcription factor like NF kappa B, which are called as the late response genes, and this NF kappa B then goes and ultimately results in the proliferation of cells. So, the entire cell proliferation program can be activated by the activation of protein kinase C, and what we will discuss in the next class is how protein kinase C can regulate gene expression by activation of both genes involved in the early response as well as late response, and how transcription factors such as CFOS and CJUN can in turn activate the genes involved in late response, leading to cell proliferation. And this protein kinase C signaling pathway is very important, because aberrant expression of these transcription factor or aberrant activation of protein kinase C can result in cancer. So, we need to understand how protein kinase C activity is regulated inside the cell, what kind of molecules actually activate protein kinase C, and what kind of transcription factors are involved in the activation of cell proliferation program, and how the entire process is regulated, and how protein kinase C plays a very important role in control of cell growth and cell differentiation. We will also discuss in very detail the mechanism by which transcription factors such as CFOS and C-Jun bind DNA, and what kind of transcription activation domains they actually possess, and what kind of residues are actually phosphorylated by protein kinase C, and when these phosphorylation, the transactivation domains, when these serine or threonine residues of CFOS or C-Jun is phosphorylated, it can result in the activation of transcription. And the very interesting thing that we are going to discuss about AP1 is that the CFOS and C-Jun, both of them contain these basic region leucine zipper proteins, and thus is very similar to the CREB, which is involved in the cyclic AMP response. So, just as protein kinase A activates CREB, which is a leucine zipper protein resulting in the activation of cyclic AMP responsive genes, in the case of protein kinase C phosphorylates proteins like CFOS and C-Jun, and as a result CFOS and C-Jun dimerize, and these dimers now go and bind to specific response elements which of genes which are involved in cell proliferation. Very interesting thing that I want to point out is that the cyclic AMP response element, which is the bound by the CREB protein, and the response elements to which the protein kinase C activated AP1 or CFOS and C-Jun bind, they are very similar. The sequence to which CREB binds is TGA CG-TCA, whereas the sequence to which the AP1 binds is TGA CTCA. There is just one nucleotide which is difference between a AP1 binding site and a CREB binding site, but when CREB gets activated by protein kinase A pathway, you have a totally different kind of physiological response. If you activate protein kinase, if you activate the AP1 through the protein kinase C pathway, you have different kind of signaling response. So, you can see there are two independent signal transduction pathways. One is protein kinase A mediated, another is protein kinase C mediated. Protein kinase A activates CREB, protein kinase C activates AP1, but ultimately at the DNA level, if you look at the sequence to which these two different kind of transcription factor binds, there is only one nucleotide which distinguishes these two elements. CREB binds TGA CG-TCA, whereas AP1 binds TGA CTCA. Instead of CG-TCA you have CTCA. So, just one nucleotide difference can bring about totally two different physiological responses, and in one case it activates the cyclic AMP response, another case it can lead to cell proliferation. So, although you have molecules which are totally diverse at the cellular level, at the molecular level you see single nucleotides or single amino acid differences can bring about totally different physiological responses and result in activation or repression of totally different kind of enzymes and proteins and so on and so forth. So, let us now try to understand the next class. Once protein kinase C is activated, what kind of transcription factors are phosphorylated protein kinase C, and what kind of physiological response is brought about by protein kinase C activation. And once we finish that, we will go back and discuss a different kind of membrane receptors, namely those membrane receptors which possess intrinsic tyrosine kinase activity, and how these tyrosine kinase receptors transduce signals through monomeric G proteins like RAS, leading to activation or repression of specific target genes. I think I will stop here.